Hi, I'm Mr. Herzon, and today on December the 15th, here's our agenda. Basically, it's the Calvin cycle. Posted in Canvas is this presentation and also a guided note sheet that you should uh, print out as you watch this presentation so you can learn from it and refer to it. And also, if you're watching this for the second or the third time, it's good to have some notes, um, a sheet of paper that is out so you can actually add what's not in the guided notes. Okay. Um, I posted some Module 5 reviews. Those are in two parts. And coming to Canvas by 6 p.m. actually turned out to be a little bit later because of the impending winter storm. It took a while to get out of Home Depot. The Calvin Cycle exit quiz has been posted, and it's just for practice. There is no grade, and you're not limited to the number of attempts on it. So it's good preparation before you actually take the Calvin Cycle quiz in class on Friday. Sorry, and then this screencast. Okay, um, Wednesday. Uh, in addition to virtual check-in, connect via Zoom for the light reactions reassessment. And then coming Friday, of course, is the Calvin Cycle quiz. The format is going to be similar to the light reaction quiz, but there won't be any questions on the leaf structure. Instead, there will be some questions from the photosynthesis bio preview. So you should review those before taking this quiz. Okay, so it will be approximately 10 multiple choice questions and 12 uh, questions in the fill in the blank essay on the Kelvin cycle. There will be a word bank, and it's worth 22 points, and it's not reassessable. And the category it goes into is all tasks and assessments. All right, we're ready to start. Well, whoops. Well, really, the Calvin cycle was first discovered by a scientist whose last name is Calvin. It's Melvin Calvin. And he used radioactive carbon and carbon dioxide. Um, Periodically, it would end up killing the cells with alcohol. Then he looked for which compounds had radioactive carbon, and this was by a timed course. So the Kelvin cycle uh, takes a period of time, okay, and he took X-ray photography of different parts of the Kelvin cycle. And he could, after developing the X ray film, he could hold it up to the light and actually count the number of carbon based compounds because of the emitting of photon light by the radioactive isotope of carbon. All right. And they would show up like little white specks on the X ray film. Calvin noted that a single carbon molecule was added in each cycle. That's CO2. And midway through the cycle, a three carbon molecule known as PGAL, or phosphoglyceraldehyde, was formed, and finally, glucose was made, disaccharides, polysaccharides, as well as some starches. That's a polysaccharide. But I was referring to, oh, amino acids. Okay. He won the Nobel Prize in Chemistry in 1961 and worked with NASA Astrobiology Division. Our own special connection here at Wootton High School is that his niece attended Wootton. Okay, so recall our rules of biology. One, follow water always moves from high to low concentration, so it's important that we follow the water. Two, substances like CO2, all right, uh, will move from high to low concentrations. Okay, anything that's permeable, um, 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 permeable um, for getting across the cell membrane moves from an area of high to low concentration. Okay, and that's in all types of passive transport. Third rule, chemical reactions, uh, for them to actually occur, you need the right stuff at the right place, at the right concentration, at the right time, right? Everything has to be there, ready to go. And once the reaction starts, okay, um, it's set to go, and it's a very dynamic process. Nothing gets used up or reinvented, and that's kind of like number four, matter cycles, and we learned that in the ecology unit. Nothing gets used up, um, and nothing gets reinvented or remade. Chemistry and the chemical reactions that occur in chemistry is all about a balance of atoms. So the reactants in photosynthesis are six molecules of CO2 and six molecules of water. Okay, we can actually count the number of atoms, and for carbon, it would be six. Okay, now there would also need to be six carbon molecules on the right side of the equation, which are the products. Okay, as well as the same number of hydrogen atoms and oxygen atoms. So on the reactants, the carbon is sustained at the number six in glucose, C6H12O6. Okay? Okay, so in photosynthesis, remember we had the light reactions, okay? There were some products in the light reactions that are needed for the Calvin cycle, which also goes by the name of carbon fixation, okay? So there's actually three parts to this. First, we're going to talk about part A, that carbon dioxide is fixed into phosphoglyceric acid, or PGA. It's a three carbon organic molecule. And, and this is done by the action of a light activated enzyme called Rubisco, all right, on RUBP. Rubisco is an acronym for ribulose biphosphate carboxylase. Now you know why we call it just Rubisco. Okay. Hydrogen ions and energy in the form of ATP transform the PGA into PGAL. Every three carbon dioxide molecules uh, uh, that are fixed forms one usable molecule of PGAL, and that stands for phosphoglyceraldehyde. Okay, so on to part A. It's important to realize that the absorption of light only happens in the light reactions. So that's the job of chlorophyll pigments A and B is to absorb the sunlight. And there it's converted into chemical energy like ATP. Okay. Now it's further going to, now next we're going to be using carbon molecules to actually harness more of that energy into the form of, say, like glucose. Okay. So the Calvin cycle, it no longer involves the capturing of sunlight. Okay. This takes place in the stroma of the chloroplast. So we look at this structural molecule of the chloroplast. You can see that it's encapsulated with um, inside of an outer membrane and an inner membrane. And then inside are these stacks, looking for my pointer here. Okay. Oh, that's kind of neat. Uh, are these stacks of membranes called thylakoids. And all of these thylakoid membranes, the entire stack is called a granum. All right. Inside of the thylakoid is a lumen. All right. So if you had a piece of hard candy, like peppermint candy, and broke it in half, okay, the outer covering of that peppermint candy would be the bilipid layer of the thylakoid membrane, okay? And when you broke it open, okay, you would see the lumen or the inside of the candy, all right? So just imagine if you had um, several of these candies stacked on one another. That would be very analogous to the granum. Okay. Now, the Calvin cycle happens inside of the stroma. It's an aqueous fluid filled with all kinds of stuff, really. Enzymes, you've got molecules of ATP, ADP, NADH, NADPH, NADP plus, as well as hydrogen ions. Okay, all right. So the light reactions produce a lot of usable form of energy called ATP. But ATP is unstable. Storing that energy only lasts for a few minutes. All right, I'll explain that in more detail. Okay, now the Calvin cycle does need light. Okay, it works the independent of the light, but light is needed, and the reason for that is that the enzyme Rubisco it only gets activated when light hits it. Okay, all right. So if see up here, here's the enzyme Rubisco. And what this is going to do is fix carbon dioxide with ribulose biphosphate, or RUBP. Okay. Now, ATP and NADPH are needed for energy to power chemical reactions. Now, these chemical reactions are mainly the altering or the changing of these carbon based compounds, rearranging them, okay, through the process. Now, um, the reason why it needs light, okay, well, if there was no light, Rubisco wouldn't be activated. And remember, ATP and NADPH, those are made in the light reactions. So if light wasn't present, we wouldn't be making any ATP and NADPH. And if there was no sunlight, remember the stomates are closed at night. Okay, so now you'll have a lower concentration of CO2 coming into the leaf. Part C is the overall chemical reaction. So we've got three molecules of carbon dioxide, okay, plus three molecules of water is going to yield, all right, one molecule of PGAL plus three molecules of oxygen. And we saw in the light reactions um, where that oxygen was evolved, and that was due to the photolysis of water into hydrogen ions and oxygen. Okay? All right, so here we go. We're going to start at like the 12 o'clock position. Okay, once again, we got Rubisco, the light activated enzyme, and we're putting in our inorganic carbon molecule, CO2. All right. Now that CO2 gets fixed, or you could, it's like the word attach, but this process or this phase is actually called carbon fixation. So here is our 5 carbon molecule of RUBP. Okay. Now, why is it biphosphate? Well, there are two phosphate molecules, one on each side. Okay. Um, Now, you just don't have one of these. Do you see this coefficient right here? That's the number 3, right? So at this period of time, there are 3 molecules of RUBP. And if we only had one carbon dioxide come in, well, we wouldn't be fixing a lot of them. So actually, there are 3 carbon dioxide molecules needed. Okay? So in the presence of Rubisco activated, we're going to make 3 of these very unstable 6 carbon molecules. They're so unstable, as soon as they form, they get rearranged because they literally just fall apart. Okay? And what forms are 6 molecules of PGA, okay, which is 3-phosphoglycerate. Now, what does the 3 stand for? Well, we number these carbon atoms. This one right here is carbon atom number 1. This one is carbon atom number 2. And this one is carbon atom number 3. So where is that phosphate located? It's located on the 3rd carbon molecule. Okay? So next is the reduction phase. Okay? And what starts this off is a molecule of ATP. All right? Energy. Now, remember, ATP is a very exists in a very short or ephemeral. It's not long lasting. Okay, so it has to release. It's unstable. It has to release that extra phosphate. Okay, so it gets reduced to ADP. Now, what happens to that phosphate molecule? Well, look up here. Remember, there was only one phosphate in PGA. Now there's a second one. Okay, that's where the phosphate goes. And since it's on the first carbon atom, now we keep track of where those 2 phosphates are by saying this is 1,3 biphosphoglycerate. The next thing that comes in, and remember that ATP and NADPH comes from the light reactions. Okay, so these are actually formed in the stroma. Okay, and what happens here is this hydrogen, right, um, gets donated and now changes the conformation of PGA to PGAL. Okay, and notice we'll have 6 of these, as well as 6 PGAs and 6 PGALs. Okay, now the reduction happens by releasing one of these um, carbon molecules from each of the PGALs. Okay, I'm sorry, from a um, one PGAL. Okay, um, gets pushed out. Okay, that's the output. So now our look what happens to our number of PGALs. We had 6 here. 3 of them are getting pushed out. I'm sorry, one of them is getting pushed out. So if you do the math, you had 6. Now you're removing 1. How many do you have left? Right, you are. Okay. We now have only 5 PGALs left. Okay. We add another phosphate. Okay. So this rearranges our molecule. And now what's going to happen is that the PGALs are actually being um, uh, joined with 2 carbon molecules, right, to make a 5th carbon molecule, a 5 carbon molecule called ribulose biphosphate. Okay, and notice we've had 3 of these. So what's happening here is that we're regenerating our UBP. Okay, so actually one PGAL comes out, while 5 are being recycled. And that goes back into the cycle. All right. Now, this makes a lot of sense because you might be thinking, well, why not use all the PGALs? Okay. Why not use all the PGALs that get generated and make glucose there? Well, if we did that, we wouldn't have anything left over to recycle our UBP. Okay? So instead of taking all of them right away, we're just going to take 1 and recycle 5, bring in 1 CO2, make our PGA, become PGAL, all right? And then 1 leaves, 5 get recycled. Okay? That's, part of the cycling of matter. Okay? So here we have our building block, PGAL, and from this we can make glucose or a whole long list of organic compounds. So these would include lipids, they would include proteins, they would include nucleic acids, all right, um, as well as other carbohydrates. Now, this is what the chemical structure of these compounds look like. Only CO2 is inorganic that you see up here. Okay. Now, here, this is PGA. Very subtle difference between this and P-gal. Okay? Um, Look at this position right here. Okay? You see there is the oxygens are in red and the hydrogens are in white. Okay? Notice here. Now look at this corresponding to PGA. All right? This part is a little bit different. Okay? And I don't know if you could see this. There's a little bit of difference right here as well. All right. Now, how are these different from each other? Okay. Phosphoglyceraldehyde and PGA. Okay. Well, here's our 5-carbon RUBP. Okay. And there's the 2 phosphates. All right. With Rubisco, the presence of Rubisco, water, and CO2, um, this is what PGA looks like. This molecule is PGA right here. All right. Now, the only difference, remember I was pointing out those hydrogens in P-gal? Those are the hydrogens that get added. Where did that hydrogen come from? That's right. It came from NADPH. Very good. Okay. So, how to remember all of this stuff? This is how we introduce finger biology. Okay. So, do you ever hear the phrase, talk to the hand? All right. You actually have 5 fingers right here on your hand. So, this is carbon atom number 1, carbon atom 2, 3, 4, and 5. Okay? On the other hand, this would be the inorganic molecule carbon dioxide. So remember, in the presence of Rubisco, that's activated by sunlight. Okay, we got the 5-carbon organic molecule RUP coming in. We add 1 carbon dioxide. Okay, remember that this is very unstable and gets rearranged into into 2 separate 3-carbon molecules, PGA. Okay, all right. Okay, now from here, remember, ATP was utilized, all right, and now we're donating the hydrogens from NADPH, and now I'm just going to flip my hands around, my palms around, and this would be PGAL, PGAL, okay? Now we're going to bank one of these. One of the carbons comes out, so now I have only left 5, and this is going to be recycled. Okay, all right. Back to the starting point. Let me make my picture a lot bigger. Okay, here we go. Calvin cycle with fingers. Okay, it's really fun to see this while students take a quiz or taking the module quiz when it comes to remembering the different stages of the Calvin cycle. You start seeing a student going like, okay, and it's a lot of joy for a teacher. Okay, so we start out, talk to the hand. All right, that's RUBP. All right. ribulose biphosphate. Okay. Now, in the presence of glucose, we're going to add one carbon from CO2. It makes this unstable um, uh, six carbon molecule that gets rearranged, falls apart to form two molecules of PGA. All right, through the help of a little bit of ATP and NADPH. Okay, we now convert that into PGAL. Okay, we're going to bank one of the carbons, and I do that by um, um, I got to start over again. Okay, RUBP, CO2 comes in, six, all right, two PGAs. Now remember, I forgot, we have to add in our ATPs and our NADPH. So that's a little bit of energy, okay? And I do the energy kind of vocal, okay? And then we have to add that um, the hydrogens. So I give that a little bzz, bzz, and then we form our PGAL. Okay, so start over once more. RUBP, CO2, reconfigure PGA. Bzz, bzz, PGAL. All right, we're going to bank one. We're going to take one away, and now we have um, 2 plus 3 is 5 to regenerate our RUBP. That's it. Okay. So here's the thing, when we bank one, that's only one. Okay. We actually have to circle. All right. Circle 3 times. All right. So we circle once, we bank one carbon. We circle twice. Now we got two. One more, we got three. That's where our PGAL gets made. That's the building block. All right. So what we would need is another three, okay, turns to make another PGAL. Now what we can do is to make our sugar molecule from that, which would require a different type of enzyme. Okay. So this is Mr. Herzon um, giving us our fist bump for the day. Is first of all, we talk to the hand, right? And we come on in with our CO2, okay? um, and this falls apart to PGA. We add some energy. And now we got PGAL. We bank one and recirculate RUBP, okay? Back to our 5, back to our hand, and there's the bump. Have a great day.